Good afternoon, everybody. Welcome back to our second session of the afternoon. In this session, we will have 3 short presentations that showcase some of the most innovative and exciting projects UK Spine has funded. We'll be looking at areas such as translational science, entrepreneurship, and bioethics. So I think it's going to be a really fascinating afternoon. So our first speaker is Ghada Alsala. Ghada is based at the Kennedy Institute of Rheumatology at Oxford, and she will be talking about the links between autophagy and healthy aging. So Ghada, thank you very much for joining us. If you would like to share your screen and take away your presentation.
Can you hear me and see my screen, everyone?
Yes, we can, Ghada.
Yeah, thank you so much. So first, thank you, David, for this nice introduction. I would like, of course, to thank UK Spine to fund my project, which, as you mentioned, David, would be about targeting or identify drug that targeting TFAB for the treatment of osteoarthritis. So osteoarthritis is the most common form of arthritis, and we know that increased aging is a primary risk factor for the development of osteoarthritis. And this has raised a great concern about the increased number of the lifespan, which is, of course, a great achievement, but with the age-related disease such as osteoarthritis, this have a great and a negative impact on the health span. With this increase of lifespan, we have an increase of about more than 60% cases of OA patients in the UK in this last 20 years, and now we achieve the number of 10 million cases in the UK. And this represent a great burden in the NHS, but also, and more important in my opinion, is that this disease affect and have a great impact on the quality of life of a patient suffering from osteoarthritis. It's prevent them to do even a very small and basic daily acts, such as opening a food container or even putting on their clothes. So this is really a serious problem, and unfortunately for the moment we didn't have any effective therapy, and most of the therapy that we have, it's most likely management the pain, because it's a very painful disease. And so we need to look at more possibility and more therapeutic treatment, because these patients most likely end up with a surgical joint replacement. What we know about osteoarthritis, that it is a disease it's a disease where we have a degradation in the cartilage, which I show you here in blue. And we know, or most of the studies showed, that actually this degradation of cartilage might be due to the senescence of cells called chondrocytes. So the chondrocytes, they are the mainly and the only cells occupy the cartilage and also maintain this tissue. But however, also osteoarthritis is considered as a whole joint disease. Many other cells besides chondrocyte participate in the development of osteoarthritis. And one of them, of course, the fibroblasts. These fibroblasts were normally situated here in the membrane synovial. And these fibroblasts also showed a senescent phenotype similar to that we have in the chondrocyte. A recent study showed that from Croft et al. It's a very interesting study, because they showed that we have actually 2 different subsets of fibroblasts, and the one which might play more role in the tissue damage is associated with osteoarthritis. However, another subset, which is have more role in the inflammation, is associated with RA, which another autoimmune disease affect also the joints. So as I mentioned to you, and actually James just have present and talk a lot about the senescence and cellular senescence in different disease. We know also that autophagy play an important role in the cartilage degradation. So autophagy, Felix, who gave his talk in the first day, gave a very nice presentation about it, so I will not go through the details, but please ask me if you have any question. So autophagy, in summary, it's a mechanism or the main mechanism of degradation and recycle in the cells, where the cells could get rid of unfolded and unwanted and damaged protein in the cells. So autophagy we were interested in our lab, and we showed that autophagy is reduced with age. And when autophagy reduced with age, could induce actually cellular senescence in different tissue, including the immune system. And recently, a very nice study showed that TFIP, which is it's a factor transcription, and TFIP is the master regulator of autophagy. When we overexpress this protein in the joint in animal model of OA, we could ameliorate the cartilage degradation. As we can see here in this figure, so here these are the sham, or they are the control mice. And then when we induce osteoarthritis using the MM model, it's a very common model for inducing arthritis. You can see here in red that we lose the cartilage. And when we inject the joint with lentivirus to increase the expression of TFIP, we resecue this cartilage degradation. And we have also similar results concerning the synovial site. In our lab in last years, we were also concentrated to understand and study TFIP. And what we found that actually TFIP is also reduced with age, and thus will lead to the decrease of autophagy and induce actually a cellular senescence in B and T cells, which have a great impact on the response to infection and also vaccination. But what I find it really fascinating and stunning that you can easily distinguish people who are 65 years old or more to those who are younger only based on the expression of TFIP and how much level of TFIP they have in their BBMCs. So I was wondering if TFIP might play a role in osteoarthritis, and if these two subsets or cell type, fibroblasts and chondrocyte, have also a reduced quantity of TFIP in OA disease. So to answer this question, I use the primary cells isolated from OA patients. They were fibroblasts and chondrocyte, and I measured TFIP expression using a FACS assay, which we developed in our lab. And as you can appreciate here, actually these cells from OA patient do have less expression of TFIP compared to our control. And on the other hand, which is really interesting, that these cells show a higher expression of beta-gal, which is one of the main markers of senescence, of course, compared to the control. But does this mean that TFIP will lead to this increase? So to answer this question, I inhibit TFIP by cRNA, and actually it was clear that when we inhibit TFIP, when we reduce the expression of TFIP, we could increase the expression of B16 and beta-gal, two main markers of the senescence. So from these data, I wondered if we could have actually identify drug which could increase and enhance the expression of TFIP in the joint, and that might be promising for the treatment of osteoarthritis. So to achieve this aim, I have two different objectives. The first one, I wanted to do a CRISPR screen to identify a regulator that regulate the expression of TFIP. And second, I wanted to do also drug screen to identify component that harnessed TFIP expression and for the treatment of osteoarthritis. So to achieve the first aim, to do a CRISPR screen, actually we did use a package of CRISPR library called the Toronto Knockout Library, V3. This library, is very it's targeting at least 18,000 human proteins, so it's most likely wild-bold CRISPR screen genome. And what I wanted to do, I wanted actually to do this experiment or this screen specifically in primary cells, and not using a cell line or other. So I extracted or isolated fibroblasts from OA patient, and you might appreciate here the increase or the huge number of cells that we need for this experiment. And to just make you imagine how much, so we have for the moment did screen in 3 different OA patients, and you can see how much like flask we need. And actually at the end, I think I became crazy. I did even get more than 200 flask for the screen. I was so excited about it. So all this flask will be infected by this library, and then we will select only the positive cells, the cells where have a package or the guide RNA in them, and then these cells were all trypsinated, and then we label for TFib and look later on for the sort to sort 3 different populations. The population with a low expression of TFib, where these might be a negative regulator for TFib, and the population which have increased TFib expression in these cells. These are a positive regulator, and of course our intermediate, where these are the neutral, or these are the guide RNA which don't have any effect. I'm so excited because my last 2 patients have been sequencing, and I have the data last week, from the sequencing, and soon we will start the analysis. However, the analysis of our first patient was very, very successful, I could say, and we have 3 very promising gene, which I hope if we confirm with the other patient, I might be able to share these results with you soon. For my second aim is to look at the drug screen. So here also I wanted to use fibroblasts, so a primary cells, where we will treat these cells with a small molecular library, and for the moment we have access to 25,000 small molecule. And of course, we will use TFib again as a readout using a high content imaging. This is in ongoing research, and we almost finished the assay development, which means you have to calculate how much cells you will need, and then how much the concentration of each drug you needed, the time, and a lot of parameter to start. And we almost did this part, and hopefully soon we could start with the primary screen. This is just to show you one of the drug, which I use it as a positive control. It's so exciting because you can see here how this drug be able to translocate TFib expression in the nucleus. So this project might be helped to identify a drug harness TFib for the treatment of osteoarthritis, but at the same time, it might be of a great benefit for other age-related disease, taking the fact that autophagy and TFib are also implicated in several age-related disease. And so, of course, at the end, I want to thank again UK Spine to fund my project. I would like to thank all my lab members, and especially Professor Katia Simon for her support. I would like to thank you very much for being here today and for bearing with me and listening to my presentation. And I'm happy to have your question.
Thank you very much, Garda. Really enjoyed that and really impressed at the scale of that CRISPR screen. There's a lot of plastic bottles going on there. May I encourage members of the audience to post questions for Garda in the chat, and we'll come back to questions at the end of the session once all 3 panelists have spoken. So thank you very much, Garda.
Thank you.
Our second speaker today is Emma Fox from the Francis Crick Institute. Emma, can I invite you to turn your camera and microphone on? So Emma will be speaking about entrepreneurial training programs that her and her colleagues have been running at the Francis Crick Institute. So Emma, over to you.
Hi, thank you, David. All right, I'll just share my screen. Okay, hopefully you can see my screen.
Yes, we can, Emma.
Okay, perfect. Okay, so many thanks for the introduction, David, and for UK Spine for inviting me today. So the main reason I'm here is to talk about translational programs that we run at the Crick around entrepreneurship. And particularly, I want to talk about the summer school program, which was a 4-day intensive program that we ran for researchers interested in entrepreneurship. So we ran it fully in person in 2019, and it was funded by UK Spine. So a huge thank you for all the support from them for funding this program. But before I go into more detail on that particular program, I first wanted to explain the background of how the summer school program came into place in the first place by giving you the context of the other entrepreneurial programs that we run out of the Crick. So first, to give you a quick introduction on the Francis Crick Institute for anyone who isn't familiar. So the Crick is an independent organization which formed in 2015, and it's dedicated to understanding the fundamental biology underlying health and disease. So it's the biggest biomedical research facility under one roof in Europe, and its founding partners are the MRC, Cancer Research UK, Wellcome, UCL, Imperial, and King's College. So I work as a program manager on the translation team, and I primarily work at the intersection where cutting edge research meets entrepreneurship, and I support health tech entrepreneurs with their ambitions and ideas. So my work is very external and ecosystem focused, particularly around the life science industrial strategy priority on data science applied to health, where Crick is the convener to bring different parts of the ecosystem together. And I work very closely with our entrepreneur in residence at the Crick, who's Barbara Domain-Heyman. So currently we run 3 external facing entrepreneurial programs annually, and they're all along the pipeline or along the trajectory from researcher to entrepreneur. So they're along different stages of that journey, our different programs. So first I'll talk about KQ Labs, which is a data-driven health accelerator, and it's our most intensive program. It runs annually between October and March and is entirely dedicated to early stage data-driven health companies. So KQ Labs started 3 years ago with the ambition and objective to help these early stage companies at the interface between data and biomedical science to get these companies investor ready and to help them raise funding following our program. So the program is run out of the Crick and funded by LifeArc, and it offers expertise and support to 10 companies that we select very carefully. And the program includes a convertible loan of £40,000. We provide weekly workshops with experts tailored to digital health. We provide training in transferable skills, mentoring by a handpicked network of experts, and we also provide introductions to investors and corporates throughout the program. So this particular program is for startups who've already had some traction, for example, pre-seed funding or an Innovate UK grant, have more than one team member. And if the founders are academics, they'll already have spun out of their universities or will be just about to. And applications for our next cohort actually open next month, and the program will run again in October. So this week, we also announced a really exciting new opportunity for early stage data-driven health companies to validate their technology through proof of concept testing and to accelerate potential adoption through participating in something called the Novartis Biome Health Hub 21 Challenge. The focus this year is on digitization of patient pathways. So the KQ Labs team is working with Novartis to help them select up to 3 winning startups, and the winning startups will benefit from mentoring, access to networks and opportunities, and also the possibility of 50,000 convertible loan from LifeArc. So the next slide here shows the breadth of impact of data-driven health across so many different areas. So these are the companies who've participated in KQ Labs in the past. So in terms of these companies and the focus on healthy aging, we do have a cluster around cancer. We also have had a company in Parkinson's disease and also 2 ophthalmology companies and 1 company focusing on remote monitoring of chronic disease. In terms of impact of the program more generally, we've seen really great progress from these companies after being part of our program. or more than half have raised significant equity funding following being part of our program, and many have won significant grants as well. So KQ Labs was really the starting point for us in terms of external entrepreneurial programs. And then we started thinking about the pipeline more widely and how we can also stimulate and support people to become more entrepreneurial from the outset. So we have another program, which is called Pulse, which is a program for up-and-coming life science entrepreneurs. This is a 3-day entrepreneurship training bootcamp, and it's aimed at all career-level researchers and first-time CEOs who are looking for practical advice, guidance, and inspiration. And we bring in lots of professionals, leading entrepreneurs, and CEOs who come and join for those 3 days. This program is run jointly with the Bioindustry Association, and applicants do need to come with a potential or an existing idea in the broadly defined fields of biotechnology, biomedical, or life science areas, as they have the opportunity at the end of the 3 days to pitch to a group of real investors and get some feedback. And so we've been running this program now for 4 years now, and we run it every March as well. And then finally, I wanted to talk about the summer school. Without the support of UK Spine, again, we really would not have been able to do this program, which was such a huge success. So in 2019, we came together with the Alan Turing Institute and also with Entrepreneur First, with the ambition of creating something to inspire budding entrepreneurs and also provide training in foundations and workable frameworks about entrepreneurship. Specifically, the objectives were to inspire data scientists and biomedical scientists to see what's possible when they come together and use their skills, experience, and insight to think about and potentially build a startup, and also around activating individuals to explore the data-driven health field further, and also to create and strengthen relationships and networks between like-minded researchers from various disciplines in the UK's top institutions, either for academic collaboration or otherwise, and also to activate individuals to explore further the field of data-driven health through academic collaboration, or by then actually applying for KQ Labs and forming new companies. So we ran the program in July 2019, and we were actually incredibly oversubscribed. We had over 300 applicants. We selected 60 data scientists and biomedical scientists from universities across the UK who came together over a 4-day bootcamp. And over the week, they had to team up with another applicant, another participant, and they had to come up with an idea over the 4 days, which they then again pitched at the end of the week to real investors. And for this program, unlike our others, applicants didn't need to be set on entrepreneurship. They didn't even need to have an idea or a team or a product in order to apply. They just had to be curious and open about new ways of thinking and about entrepreneurship. And so for researchers with entrepreneurial ambitions, it was a good opportunity to learn some foundations and workable frameworks about entrepreneurship. And again, UK Spine was really crucial in helping us make this program happen. So we had a really great result from this particular program. So we had 2 researchers who came together during the summer school with the idea of a data-driven vaccine design platform. A few months later, they actually applied to our KQ Labs Data-Driven Health Accelerator program and were accepted. And then also their real success story, because just a few months ago, they recently announced that they were able to close a seed fundraising round to start developing a variant-proof COVID vaccine, which is very topical, and we're really proud of them as well. So this example really shows the potential impact of a program like the summer school to activate people to entrepreneurship and think how they can make an impact with their science. So due to COVID-19 in 2020, we weren't able to do an in-person summer school, but we did do a series of virtual masterclasses, so some panels and workshops that we did virtually. And this year, our plans are still in progress. So COVID is still causing a lot of uncertainty because the nature of the kind of event, at least the one we did in 2019, meant that the original format was very interactive, and there was lots of informal networking to get people to create teams over 4 days. So it's unlikely that we'll be able to do it in its original format in person, taking into account safety and how things will go in terms of COVID. However, we are thinking about our plans for this summer very much in the next few weeks and deciding what we do want to do this year. So we'll come to a decision quite soon. So do watch this space. And then just wanted to close off by saying another huge thank you to UK Spine for having me and for all their support and working with us in that intersection between cutting edge research and entrepreneurship, and in inspiring and training budding entrepreneurs. And yeah, I believe we'll have some time at the end for some questions.
Thank you very much, Emma. That was a really great talk. And I think that Basinine story is really inspiring about what can be done when you get the right people with the right training.
Exactly.
So I'd now like to invite Heather Draper to join us on the stage. Heather, if you could turn on your camera and microphone. Heather is the chair of bioethics at Warwick Medical School, and she will today be talking about a concept of donor-funded clinical trials. So Heather, I'd invite you to share your slides and begin your presentation.
Hi. I'd also like to thank UK Spine for funding this project. I'm actually presenting a whole project as quickly as I can. This was the title of our project, and the investigators are listed, and also I've mentioned Kirsty Sherman, who's been really helpful and was additionally funded to help us through the COVID bump. It would seem I have to be on my slides. Okay, so the aim of our project was to explore the potential barriers to developing an ethically acceptable set of research protocols for donor-funded clinical trials, and also to work out whether these barriers could in principle be overcome to enable researchers to secure a favourable REC review, given that this was a novel funding stream. Some of you may have heard Alexander talk about the funding model last year, but in case you didn't, I'm going to first briefly describe this model, and then I'm going to talk briefly about how we did the project, and then go into a description of our findings. So this is the model that we were looking at. So the starting assumption is that there is insufficient funds available to fund all promising agents. And there are also extremely wealthy people who are looking for trials to help with the conditions that they are experiencing. So the idea is that there's a matching agency who brings together donors who have huge amounts of money at their disposal and researchers who have promising clinical trials they would like to have funded. Now, they don't take it at face value that the researchers have promising research. The idea is that there'll be expert review of their research protocols. And when everyone is satisfied that there is a promising agent and a good design of the clinical trial, the funding for that trial will be agreed. And it will be then down to the researchers to gain the approvals, for example, the REC approval, before setting up the trial. But the important thing about this model is that the donor is guaranteed a place on the clinical trial, provided they meet the inclusion and exclusion criteria at the point of recruitment. The funding works like this. The donor lodges the money with the agency once the project's been agreed. And this is a bit like a solicitor when there's an exchange of housing contracts when you're buying a house. So no matter what the outcome, no matter whether the donor is still alive at the point that the trial begins to run, the funding is secure and safe for the researchers. And the matching agency may withhold a bit of the funding to ensure that the researchers complete the project and publish their results, which is not unusual in the way that funding agencies work. And if you're interested in reading about the entire committed philanthropy model, the reference is there on the screen. So our method in brief was to take an empirical ethics approach. That is to say, we thought that it wasn't good enough to simply do an ethical analysis of what the issues were. We also had to see how acceptable our ideas were, and with the relevant stakeholders, and also to get their perspectives on this type of funding so that we could address those. We thought that the three pertinent stakeholders for this type of funding were REC members themselves, and we chose REC chairs as a proxy for them as they had the most experience. Potential other people who might be participants in clinical trials, and for this we used PPI group members of clinical trials unit and also the researchers. We held 3 focus groups eventually online last year in September, October and December, and there were 22 participants in total. Now, normally when I'm presenting the results of qualitative research, I would be talking in quite a lot of detail about what the specific participants said and how they said it, and trying to explain how I interpreted the data. But given the 5-slide restriction and 15 minutes to present, that hasn't really been possible. So I'll just present the 7 overarching main themes. So the first theme that emerged only from the researcher group was that this model of donor funding threatens to disrupt in a bad way the research agenda and threatens research infrastructure. So just to explain a little bit about what this was taken to mean, the researchers were concerned, or some of the researchers were concerned, that normally funding is channeled towards areas where maximum benefit can be achieved. And because the donor is only interested in whatever particularly ails them or their family member, this may not be something that will help the majority. So the research agenda may be skewed in a way towards the donor's interest rather than the general good. And the infrastructure point was raised to meet the idea that, but donor funding's bringing additional money in, so how can it be disrupting the agenda? Surely it's enhancing the agenda. It was pointed out that the infrastructure itself is a limited resource, and researchers and laboratory capacity could be pulled towards that project. So that was only a theme discussed in the researcher group. The second theme that was predominantly discussed in the PPI group, but also was raised in some of my pilot work in advance of this project, is the idea that research, even good research, even valuable research, shouldn't be funded with what was thought of as dirty money, or indeed by corrupt individuals. So there were concerns that the source of funding mattered, and the people mattered. There was concerns that reputational damage could be forthcoming if bad people were known to have funded research or researchers or institutions. Now, not everyone held this view, but where it was held very strongly. Other people felt that normally we don't know the source of funding for research, and that this was too high a criteria for a funding body to reach, that all its money was clean. But people who did hold this view did hold it quite strongly and continued to hold it throughout the focus group. The third theme was how the matching agency would be operating and it would be governed and how it would be funded. So there were concerns, for example, around how it would be sure that it got the right experts to review the research protocols. And it was understandable that people wanted to know more about this matching agency, because it's a bit unknown and unfamiliar quantity, because this is a novel view of funding. People also wanted to know how the matching agency itself would be funded, and how it could be sure of being sustainable, since it had such a key role in the flow of funds from the donor to the researcher. By and large, people were advocating for the agency to run very much along traditional lines of a funding body, which in our user group, which we held after the focus groups, one user pointed out meant that they were just trying to kind of squash this model back into the traditional model and bring it into line, as it were. So they didn't like the concerns that had been expressed there. The fourth theme, and this was a theme that came up in every single one of the focus groups, was the idea that it was very important that any science that happened or any clinical trials that happened were scientifically robust. And this was linked very strongly to ethics. For example, one participant said, "We know that good science is good research and that it's good ethics." So this was feeding off the kind of quite well known mantra in research ethics that bad science is bad ethics. There were concerns that the standard for good science was expert peer review, and also that there should be a PPI element to trials. And people were very concerned that charismatic, particularly in the researcher group, that charismatic researchers who were overconfident about the product that they had could potentially persuade donors to fund all kinds of crackpot, and I'm using that as a quote from one of the participants, crackpot products or wacky products, and the whole Trump and bleach saga, because it was a contemporary issue, was raised as an example of how this could happen. So everyone thought that robust science was very important, and this was a very strongly held view. But I think how it was presented was that the donor should not be able to influence the trial design. And I think for reasons that I'm happy to go into in questions, that there's a little bit of tension here between the sense that the donor can influence the trial design and the criteria for robust science. The fifth area of concern was the rather meaty area of the donor being guaranteed this place on the trial. And this raised three areas of concern. The first was what was the impact of the donor actually being identified as the source of funding. And there were various concerns around this to do with protection of the donor's privacy, but also the potential of an identifiable donor to influence how a trial ran, or to potentially have a biasing influence on the results. There were also concerns expressed for the donor themselves. There were concerns expressed that the donor may suffer from what's called the therapeutic misconception that the research would actually definitely benefit them, whereas, of course, research is research and no one can definitely benefit, both at the point of funding and at the point of being a participant. But the big range of concerns were around fairness, and there was a concern that it had been a kind of secure principle of clinical trials that research participation was never premised on the ability to pay, and that this would somehow undermine this very important and noble premise. Other people, however, felt that the world was an unfair place, and the rich in many ways used their wealth to secure benefit for themselves, and that this was no different to that. So we're less concerned about fairness in principle here, although obviously they understood what everyone else was saying. There was a further line of reasoning around the additional funding and whether additional donors could be added. And this is a problem really because of the balance of donors to non-funder participants, which I haven't really got time to go into. And the seventh theme arose because I asked every group at the end whether they had changed their views over the course of the focus group and what they generally thought now about this model of funding. And by and large, participants didn't have any major problems. Some of them completely endorsed the idea. Some of them endorsed it with caveats or had residual concerns they would have liked to have seen addressed. And only one person really steadfastly remained against the notion of donor funding. In the REC group, there was a consensus that donor funding didn't raise such peculiar issues that it required a kind of special category of research of REC to review it. So I don't know whether you're aware that there are recs that are designated for children or for mental capacity issues. They didn't think that that would be necessary. They thought it should be looked at as a mainstream proposal, but they also thought that guidance on the issue would be helpful, at least at the beginning. So what did we, or how did we organise these themes? We looked at those themes and thought that they needed to be presented in a particular kind of way, and this is how we've worked through it. We felt that they were issues that were raised that were actually outside of the scope of ethics review, and that is the scope of the review as defined by HRA policy. And these were dirty money, disruption to the research agenda and governance of the matching agency. We felt that these were issues that the REC wouldn't normally get involved with, wouldn't normally receive information about, so it was difficult to justify why this information would be pertinent for this particular source of funding. And then there were issues that should already be covered by a standard REC review. And this included the concerns around the robustness of the trial. So RECs would normally get expert reviews such as that the researchers already had. They would have access to those, for instance, the ones through the funding bodies. And if they're not satisfied with the reviews they have, they are actually able to request additional reviews. We also felt that funding was an area of REC concern and that this would be something that we could provide guidance on, but it wasn't a specific issue. So the central issues of concern were the concerns around the donor securing the place on the trial. We think that it probably is advisable for the donor to be kept anonymous for reasons that I can explain and that would need to be explained in the guidance. We agreed that it was important that the donor understood the nature of research and that there could be measures put in place to ensure that RECs could be satisfied about this, and that of course it's down to RECs, because it's one of their central roles, to ensure that the burdens and benefits are fairly distributed. So we have our results now. We know what we're going to do with them. We've run them past our user panel, and so far it seems as though the issues as they affect RECs are surmountable. There are challenges that remain. We need a matching agency, or rather people who are interested in this funding model need a matching agency. And obviously this is going to have some kind of perhaps online presence so that people can be reassured about transparency and clarity as regard to its operations. We are intending to publish our findings. Our paper is in a good state of repair now. And we're also going to attach to this paper the beginnings of suggested guidance that can then become an evolving document. Actually, as a result of the interaction with the user panel, we've now been asked to get in touch with the HRA, who are very interested to hear what we're doing. And the next step will be for Dominic and Alexander to take the project forward and try and secure funds for a specific project, and then we can truly see whether the guidance and the perceptions of our user panel will help them to prepare their application for REC funding. So thank you again to UK Spine for funding the project, and I'll look forward to your questions at the end.
Thank you very much, Heather. That was really, really fascinating to get people's insights on this question. May I invite Garda and Emma to join us back on the stage, please? So if you turn on your camera and microphone, and we have some questions that have come in. So the first question I have here is for Garda. So Garda, towards the end of your presentation, you noted that TFEB pathway influences numerous age-associated morbidities. So with your work focusing on osteoarthritis biosamples, is there a path to expanding the impact of this work to identify interventions for other age-associated autophagy decline, particularly related to multiple morbidities?
Yeah, thank you very much. This is a very great question. I think in my project, there are two possibilities to accelerate that, and this is something we are very keen on, actually. So as the CRISPR the drug screen would be in somehow also using some library where are repurpose of the drug. So in these cases, if one of these drug were successful to increase the Fab expression, so we could actually go rapidly via clinical trial in a small group in different category. So this is some of the possibility. The other one, if it's a new drug where we don't know about the safety, and of course, in any way, we need to go through the animal validation of our, to validate our targeted drug, or even from the CRISPR screen, we need to use some animal model. And in this part, we could also induce or validate this target not only in an animal model of osteoarthritis. We could use other regenerative, like other age-related disease or morbidity. So this is possible, and something we also project in to be able to do what is the next step, what is the best to do. Yeah.
Thank you, Garda. That's really interesting, and I think touches on some of the themes of the last talks we had from James and Graham as well about repurposing. The next question I've got is for Emma. So Emma, patients tend to be very positive about working with the NHS and providing their data to the NHS, but they can be quite hesitant where private companies are involved. So on your program, is there training for transparency and communication with potential users of patients' data?
Yes, thanks for the question, and it is a very important issue. So we do introduce the startups to Health Data Research UK and Genomics England, and we also do actually conduct a session on data ethics. So we brought in the Open Data Institute in the past, who did a really great interactive session with a few hours of training on their data ethics canvas, looking at exactly those kind of issues around transparency and communication with potential users. We also have a session around data and IT security. But again, at the end of the day, it is up to the companies. However, we do the best that we can to support them to make those right decisions in terms of data security and communication, as you said.
Thank you very much, Emma. So the next question I have in is for Heather. So Heather, you talked about one of the concerns raised about the donor-funded trials method was that it might disrupt the current way that research is done and funded. But just lost my question off the screen. Many in research would see the current funding cycle themselves as having a distorting effect, the distorting effect of grant applications and peer review. So was that considered and explored with the group about how the new distortions caused by donor funding might be different from old distortions caused by the current methods of funding trials?
I didn't argue back very much with anyone when I ran the focus groups, because my starting position on this was that I was neutral towards donor funding. That's why I've been brought in to run the project. And I didn't see it was my place to argue vociferously for it. I did put it to the researchers that people were already saying that the research agenda was well, there's the 90-10 agenda, isn't there, that 90% of the world's resources go to 10% of the population. So I think there is a general question about whether this research agenda is as noble and pure as people perhaps think it is. And I think that some of the contributors to that session asked everyone if they wanted to do this, if they wanted to send further comments to do so. And one person felt that the people who felt the most strongly about this were people who were already quite key figures in research and were maybe very successful at getting funding, and just didn't see that there were many good ideas that didn't get funded, and that the agenda is in some respects skewed towards the successful. So I think that I didn't personally argue back with them, but that doesn't mean that because of the model we're taking this, the method we're using of empirical bioethics, I am able to take into account that there are counter arguments and present those alongside the views of the participants. Don't know if that helps to answer Leah's question.
No, I think that's a really great answer. Thank you very much. So the next question I've got is for Emma. So Emma, are there common factors which seem to discourage academic scientists from exploring entrepreneurial opportunities?
Yeah, thanks, David. I would say that when we started our summer school, we were quite careful with the positioning that we took when advertising and promoting it, to be actually talking more around the impact that you could make with your science in terms of, instead of really going quite, aggressively on the entrepreneurship angle. And we found that we had quite a few people who then joined the program, not knowing it was going to be as much about entrepreneurship, but saying that they were so glad that they had joined, and it really opened their eyes to a path that they hadn't really thought about in the past. So I think there are some common themes of people perhaps not knowing what the first step is. A common question perhaps is, I have an idea, but I don't quite know what I should do with it, or I don't know if I'm the right person to run a business. I also think perhaps there's the busyness of being a PhD or postdoc, and the types of pressures that you have to publish papers and so on. And then I think also perhaps just not being exposed to entrepreneurship as a possible career option. So I think that's why these kind of programs right at the early stages are important, because they really sow the seeds for then getting those companies into the ecosystem. So without that early stage fostering of entrepreneurship, then you can't have the later stages. So, yeah.
Thank you very much. So just a very quick follow-up on that. Do you ever get people who go on your program and come out saying, no, I've decided entrepreneurship isn't for me?
Yes, we have had a couple as well in that way, but I think it's more of, I would say that even them have said that it's really opened my eyes to what's possible. And I think also, maybe it's not the right thing right now, but it could be in a few years. You think, actually, maybe now's the right time. So I think, yeah, we have had it on both sides. And I think it also you don't have to leave academia and just become an entrepreneur. There are lots of successful academics who, are part of companies, but are primarily still academics. So I think they're also seeing the breadth of possibility and options is also, I think, important.
Thank you very much. So the next question I have is for Garda. I think this is a very interesting one. So is reduced TFEB associated with any other conditions, which is sort of the first part of it? And then are there any drawbacks of potentially targeting TFEB? So for example, is overexpression associated with any cancers? You're on mute, Garda.
Sorry. So the first part was, because I can't see the question. So the first one was if a TFEB reduction is associated with other disease. Is that right? Yeah, actually it have been studied in different, such as Parkinson disease, and also in also the lysosome disorder disease, Huntington disease. So a lot actually of age-related disease have been shown in different kind of cells that TFEB were reduced in this model. What was the second one, sorry? So
if you then increase TFEB expression, are there any dangers of pushing it up too high, for example, cancers?
Yeah, of course. Of course, we know that actually autophagy and TFEB might be, some people want to target and inhibit it in cancer, in specific cancer situation. So I do believe that everything needs to have a balance. So if you have a normal expression of TFEB and you come to have a more higher of this protein, I think here we will have more, we again playing with the balance of this protein, and so then we will have this side effect and unwanted effect in somehow. But however, if you have a lower expression, which is not normal, and now we want to increase that, then we go to the normal. We want to be back to the normal balance. So this will not be a problem in my opinion. But it's always also, if you find the drug, you have to work in the dosage, the time, and it's a lot of course, parameter may play in this situation.
Thank you very much, Garda. So what I think will be our final question of the session is for Heather. So Heather, based on sort of progress observed to date, what would the anticipated remaining work be, and what is the realistic time frame for a first UK funded trial via this donor funded method? Oh, you're on mute, Heather.
Probably just as well, because at that particular moment I was waffling. The end date for us is quite close. I guess it will, some may depend on how involved the HRA gets. They weren't particularly interested when at the start of the year because of the COVID problems. But I think that it also depends how quickly Dominic and Alexander could raise money. But I would have thought, given their past performance, maybe 3 years. But I'm only guessing. Maybe ask Alexander in the next session, Monica.
There you go. That's maybe 3 years will be in time for Garda to have one of her drugs from her drug screen ready to go into one of those trials.
Well, I was wondering whether Emma, whether your company, Emma, or your group, Emma, scrutinizes the sources of funding, and whether there are these concerns around clean money, dirty money, nice people, bad people, when your people are pitching, when your entrepreneurs are pitching.
Yes, I think looking at trying to find a good investor, I think, is a really important thing that we look at, is that if you have the possibility, you want to try and find someone who you also ideally get on with, because especially for the kinds of companies, for us, if you have an investor, they might be, with you for 10 years. So ideally you'd want someone also that, that you have a better relationship with. And that it's very much something we look at when we mentor our companies, is getting the right kind of investment and funding, if at all possible.
There you go. Thank you very much to all three of our speakers from this session again. I really enjoyed listening to your work. We now have a half-hour break before our final panel session. I would encourage people to get out and have a little walk around in the sunshine, have a glass of water, but also to make use of the networking and chat features of Hopin platform. If you click on networking on the left-hand side of your screen, you will be matched with another delegate who you can have a conversation with, which recreates the real-life conference experience. And you can also use the chat function on the side of the page to talk to other delegates, or you can start one-to-one text chats with other delegates who you might have seen pop up and you want to talk to them about the questions they've raised. So with that, I will close this session. Thank you very much to Garda, Emma, and Heather again, and I will see you all again in half an hour.
Thank you, David. Bye.